Hello everyone, welcome back to SLD Biology. Let us now proceed with the excretion part 2, and in this video we will mainly focus on how various parts of nephron are adapted to carry out their functions. Let us firstly proceed with the glomerular capillaries, which are a network of tiny blood capillaries in the Bowman's capsule of a nephron. The walls of the glomerular capillaries, they contain tiny pores called fenestrations, as you can see here. So these are the tiny pores. And these tiny pores are highly permeable to water and small solutes. They will allow them to pass through the pores easily during ultrafiltration. So you see all these small solutes, such as nitrogenous waste, water molecules, amino acids, glucose, they get filtered. However, fenestrations are restrictive to larger molecules such as proteins and blood cells. They will prevent their passage into the filtrate. But the thing is, fenestrations somehow allow most other constituents to pass through. So that is why we need to have an additional barrier, which is basement membrane. So the basement membrane lies between the endothelial cells of the glomerular capillaries and inner layer or visceral layer of Bowman's capsule, and they act as a size and charge selective barrier. The glomerular basement membrane, it acts as a physical barrier. It will prevent large molecules from passing through, and since basement membrane is negatively charged, they will repel the similarly charged, negatively charged plasma proteins, example albumin, and they will prevent their filtration. And basement membrane will also provide stability to the capillary network and help maintain the shape of the filtration barrier. Therefore, keep in mind that fenestrations, which are pores in the endothelial cells of the glomerular capillaries, although they prevent the filtration of larger components, they will allow all other plasma constituents to pass through, so they are not highly selective. But when it comes to the basement membrane, they are highly selective. They will prevent the filtration of those larger proteins. So together they act as ultrafilters in the glomerular capillaries. When it comes to the inner layer or visceral layer of Bowman's capsule, they consist of a specialized cells. There are many such cells called podocytes. So these are the podocyte cells. And these podocytes, they have feet-like extensions called pedicels, which will wrap around the glomerular capillaries. And this will result in the formation of a filtration slits, which are highly selective. So these filtration slits in the Bowman's capsule, especially the visceral layer, will prevent the essential loss of cells and large proteins from the blood. It gets retained in the blood, and only the small molecules such as solutes, including the nitrogenous waste ions, excess solutes, and water are being filtered from the filtration slits. So to sum up, fenestrated endothelial of the glomerulus, glomerular basement membrane, which is size and charge selective barrier, and visceral layer of Bowman's capsule consisting of podocytes with filtration slits. Together, these three layers forms an ultrafilters or ultrafiltration barrier, which will allow only the selective filtration of the blood plasma. It will allow waste products and excess substances to pass into the nephron and forms the filtrate, while it will retain essential proteins and cells in the bloodstream. Moving on with the proximal convoluted tubule, which plays critical role in the reabsorption of majority of the substances, around 65 to 70% of the filtrate. They enter back into the blood by exiting the filtrate. So if you see here, majority of the water, ions such as sodium, chloride, potassium, etc., they enter back into the blood, indicating that majority of these substances, they have gotten reabsorbed. So now what are the features that allow this major reabsorption? So let us move on with the features, that is structural adaptations that maximizes the efficiency of this selective reabsorption process. If you look at the inner surface of the PCT cells, it is covered with microvilli and it gives the appearance of a brush bordered. So these microvilli significantly increases the surface area for absorption. That gives the maximum reabsorption in PCT. And not only this, they also contain a high density of mitochondria, which provides ATP required for the active transport mechanisms involved in selective reabsorption. And they also contain many transport protein channels which are needed to absorb glucose and amino acids and ions. And they also contain which enables the active transport of the substances. So together, these proteins, transport protein channels and sodium potassium pump, they enable both passive and active transport, and that is how they ensure that majority of the valuable molecules are reabsorbed efficiently, while waste products and excess substances, they remain in the filtrate. And not to forget, you will get to see tight junction label there in the diagram. These tight junctions, they will prevent the leakage of substances between the cells, and they will ensure that reabsorbed molecules are directed into the interstitial space there, and subsequently they will enter the peritubular capillaries for the reabsorption into the bloodstream. PCT also plays a vital role in the regulation of blood pH. Moving on with the structural adaptation of the loop of Henle for water reabsorption, the thin descending limb is highly permeable to water but impermeable to salts, and you can see the water exiting the filtrate, and it will exit entirely by osmosis, and it will enter the interstitial medullary fluid and ultimately into the vasa recta. As you can see there, the water exiting the descending limb will enter the bloodstream, that is into the vasa recta. Regarding the ascending limb, the entire length of the ascending limb is impermeable to water, preventing further loss of water, and they are highly permeable to solutes. So which means they will allow solutes to be removed from the filtrate into the interstitial medullary fluid, and if needed, they can enter the bloodstream. This difference in properties of descending and ascending limb results in different tonicity. The filtrate inside the descending limb becomes hypertonic, and in ascending limb, it results in hypotonic. The next thing we are going to discuss is the length of loop of Henle. As you can see clearly in the diagram, the length of loop of Henle differs. Some loop of Henle are of short length and some they have long length. So the loop of Henle with the short length is known as cortical nephrons, and the loop of Henle with long length is known as juxtamedullary nephrons. Cortical nephrons with the short loop of Henle, they are mostly found in the cortex region of the kidney. So because of their short length of loop of Henle, they are not able to create strong osmotic gradient in the kidney medulla because they don't reach there. So you see, in order to reabsorb or take part in water reabsorption, they need to have longer loop of Henle. Then only they can create this strong osmotic gradient. So that is the key reason why cortical nephrons cannot participate in concentrating the urine and reabsorption of water. So the cortical nephrons are mostly found in aquatic animals. Because those animals who live in water-rich environments, water conservation isn't much of a concern. These animals, they don't need to produce highly concentrated urine, and they don't need to conserve water. So that is why these cortical nephrons are found in animals that live in aquatic environment. While juxtamedullary nephron with the longer loop of Henle, they extend deeper into the kidney medulla, and they are able to take part in the creation of a strong osmotic gradient, which is very required or so required in producing the concentrated urine and conserving the water. So that is why animals with juxtamedullary nephrons are typically those that need to conserve water, and they are especially found in arid or dry environments where the water is scarce. So the long loop of Henle helps them reabsorb more water from the filtrate, making their urine more concentrated, and this helps them or prevents them from getting dehydrated. And those animals living in normal conditions, including humans, they will have both cortical and juxtamedullary nephrons, but compared to juxtamedullary nephrons, they will have more of cortical nephrons. The reason why they have both is to balance the water need, and the reason why they have more of cortical nephron is because they are exposed to relatively accessible water. Not to forget, loop of Henle also takes part in countercurrent mechanism, which I'll be making a separate video lesson on it. Now you can try answering some of the questions that I'm going to present here. Write these questions down. You can pause it, copy in your notebook, and try to answer these questions. If you have any doubt, leave it in the comment section. And see you in the next video. If you haven't subscribed yet, please do so. Take care. See you in the next video.